Hi everyone, so today I'm just going to kind of quickly demonstrate how we can go from a strand of RNA, DNA, anticodon, and keep all the 5' to 3' directions good, and how you can just start with one of those sequences and you can build the entire molecule. So the big principle that we've found with these nucleotides, anything like that, is that they run 5' to 3' in a single sequence. You can't ever have them aligning in the same direction. You can't be parallel. The nucleotides basically miss each other and they don't bind up. So this isn't a possible conformation. Everything that we do, thus, is going to run anti-parallel. All the connections we make, so any A's to T's, G's to C's, they all have to be if we're running anti-parallel. And that's what this idea is. So, if we start with an mRNA, and let's say we're going to do U, G, C. So we're in this kind of forward direction that we're reading it. First thing we can do is actually, if say we're starting with mRNA, we can read this for the amino acid that it's going to code for. So U, G, C. This actually equals a cysteine. So that's the first thing you can do with the RNA, is you can immediately tell, using your genetic code, which you'll have on the test, which amino acid it's going to code for in this longer sequence. Next thing, let's take a look at the DNA that this came from. So if this was binding DNA, and this is in 5' to 3', that means that the DNA that this was built off of had to run the other way. It had to run like that. See how we're going to make this connection up to DNA? Okay. And as long as you keep the strands anti-parallel, you can just match everything up as you go. In this case, that means that this strand, but remember this is 5' to 3', but that doesn't matter as long as you tell me the direction, A, C, and G. That means that the other strand of DNA is always going to run opposite, just like this. That means in the DNA world, and in this helix right here, basically, we have T, G, C. So that's why each of these DNA molecules gets a different name. This one, so on the bottom, is the template strand, because you're actually building your RNA right here off of these three nucleotides. And remember, this is going to happen for a lot larger than just 3, but we're just doing this example with 3. On the bottom, or on the top in this case, is the coding. And that's because these nucleotides, although they're not going to physically interact or make the RNA, they look like the RNA because they're running in the right direction, same way as the RNA. So G, G, C, C. But here you see that, remember, T shows up in DNA, but U is in RNA. It's the equivalent in RNA. Okay, so from this I could actually give you, let's say I gave you the template strand of DNA. You could start here, make a coding strand of DNA. You could make an mRNA out of your template strand. So that may take you kind of reversing the order so you can read it well, but as long as I present it in a certain direction, you should be okay. So the last thing that's going to correspond here, so we have our protein, but the way we actually get to our protein is from the anticodon. Because remember, this is a codon for RNA. Anticodon comes from your tRNAs, and these are actually what's bound to cysteine. So what's the cysteine attached tRNA for this? All you have to do is, again, remember that this is this direction, 5' to 3'. So all we have to do is design an anti-parallel strand, 5' to 3' for the tRNA anticodon. And as long as you're facing this direction like that, you can just translate directly. A, C, G. So on the big tRNA, so that big goofy shaped thing, this is the codon part, that little loop that's directly going to interact with the RNA, and this is directly attached upwards in the tRNA molecule itself with this cysteine. So that's your brief overview of how I can give you any of these molecules and a direction, and you'll be able to give me this entire picture from anything that I give you. Now, I can't just give you the protein, because, for example, cysteine, what we just did, could be UGC mRNA, or it could be UGU. So that's the one thing that I can't give you, is just say like, oh, this amino acid's here, unless it's one of the amino acids that only has one working triplicate.